Today we're going to talk about medical school histology, images of the human testis. You'll be able to download these images. We have 3 of them. One is H&E or hematoxin and eosin, and the other 2 are toluidine and blue stained tissues. You'll be able to identify the different cells in the testis, the different germ cells, spermatogonia, spermatocytes, and spermatids, and the characteristics of the interstitium, as well as the capsule of the testis. Enjoy. Medical School Histology, Images of the Human Testis. Hi, I'm Larry Johnson, a professor at Texas A&M University, and you're on my YouTube site, VIBS Histology. Please subscribe if this information is useful. Today you'll be able to observe and manipulate the images that we're speaking of today. There's 3 of these images. One is stained with H&E or hematoxin and eosin, and 2 are stained with toluidine and blue. Everything is blue. Different intensities indicate different structures that are there and shapes of the structures. So you'll be able to go to these links, download, observe, manipulate the images yourself. So the male reproductive system is composed of various organs, but today we're going to talk about the testis. We'll see the epididymis, but basically we're talking about the testis, its capsule, the blood vessels that run around inside the capsule that facilitate cooling of the testis. And here we can see the testis here, and the inside we can see the seminifertubules. These are the tubes that produce the sperm, and we can see the tunica albuginea, or the white coat, reflected back. So this is the capsule that we see around through there, reflected back, and here's the epididymis in through there, and then the ductus deferens, this is the spermatic cord, in through here. Now the main product of spermatogenesis is the spermatozoa. And here we can see a human spermatozoan. There's its head, there's its middle piece of the tail, mitochondria around the tail, and acrosomal contents and membranes located at the apical end of the head. We can see epididymal sperm in through here. There's efferent ducts, but this is epididymis, and we can see sperm in through their little bullets. If we look at it more closely with the stallion spermatozoa, and you can see the nucleus and a host of membranes around and associated with the acrosome, and development of the tail with the different components of the tail as you come down its length. So the main product of spermatogenesis in the testis is to produce gametes for fertilization. First slide that we're going to look at shows the capsule. Like here we see the capsule. We see blood vessels in through here. These are blood vessels. These are arteries and veins that we see. You can see valves in the veins that we see through there. Also the arteries could be seen throughout there. You can also see lymphatics. And here we can see a lymphatic with it has a valve in it as well. And then we can identify the different cells in the seminifertubules. And if these are the myoid cells, tubule, there's a nucleus of one there, there's another nucleus here. These are the myoid cells that surround the tubule and mark the limit, the outer limit of the tubule. Inside here we have these Sertoli cells, which are nurse cells, and we see one here, see one there, there, and here's a very nice one here. They're kind of pear-shaped and a bit of a big nucleolus. Those are support cells that extend from the base to the lumen, and all the germ cells are kind of in the pockets, not inside, but in the pocket of the little membrane-bound containers that the germ cells are in between the Sertoli cells. But we can see the different cells there. The spermatogonia are located in the base, so you have the most immature cells here and the most advanced in the lumen where they're going to be released. So we have the different types of spermatogonia. We have the pale, we have the dark, 2 different types, and we have primary spermatocytes. Here is the early primary spermatocytes, and these are late or Pachytene primary spermatocytes. So largest nucleus in there is going to be a Pachytene primary spermatocyte. But other spermatocytes are smaller as well, but they're off the base, a little bit in from where the spermatogonia are located. Next, we can see secondary spermatocytes. So these pachytene primary spermatocytes divide, and we can see more of those here to give you secondary spermatocytes. Secondary spermatocytes divide to give you spermatids. And so we can see these spermatids in through here, here, and here. These are the ones that are spherical ones, and then whenever they elongate, they become bullet-shaped like sperm. So these are really spermatids as well. The mature spermatids are just about to be released or spermiated. So we can identify these cells. Here's the Sertoli cells, as we mentioned before. And if we look at the first one of these images, we can see that here we can see the capsule of the testis, and we can see the seminal tubules. So these are the different tubules are located here. I can just click and drag, and I can able to see the different structures and manipulate the structures. I'm rolling it here to change magnification, or you can change the magnification at the top. So here we see the capsule, the tunicule albuginea, or the white coat, and here we see mesenchymal cells located on the surface. So these are epithelial cells located in here to allow the testis to kind of slide up and down within its container. And so then we can see there's blood vessels in through there. These are the veins, and these are the arteries. Arteries right in through there and there. And we can see also there's lymphatics. So if this is a vein, you can see the endothelial cells lining the valve. So that's a vein. And then these are endothelial cells lining a lymphatic. The testes were perfused, fixed, and in doing so, it washed out the blood. So where this would be blood is all gone. However, the perfusion through the vasculature does not remove the lymph. So this is the lymph that's located in there, and so we can see that's a lymphatic as opposed to veins or arteriole that we see there. Also, we can see there's nerve. This is nervous tissue. These are Schwann cells in through there, and myelinated axons located in through there. And sometimes you can see if you follow the connective tissue going through here, we can see the connective tissue going through there. Usually there's nerves, not that many, but there are some nerves in through there, as well as the blood vessels kind of follow the connective tissue going in through there. So if we look at the interstitium inside there, we can see there's a series of these tubes. These are the seminal tubules, and these are the Leydig cells. These are Leydig cells right through there, and there's a little, probably arteriole in through there, maybe a venule. But if we look at a higher mag, we can see the different cells. You can see the myoid cell, there's a nucleus of it, but they surround the tubule. And we can see the Sertoli cells, the pear-shaped Sertoli cells with a big nucleolus, or these guys here, 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 are Sertoli cells. Spermatogonia are located in through here, and these are spermatocytes. This is a spermatocyte. This is a later spermatocyte. So as you advance in age, you go toward the lumen. And then these are spherical spermatids, and these are elongating spermatids. So the location at the base or the lumen indicates what cell type it is, but also there's characteristics of the nucleus. The nucleus may be oval or may be round. In the case of spermatogonia, they will be spherical in the case of spermatocytes and spermatids until they start to elongate. And so we can see these cells. These are interstitial cells. There's one cell here and another cell here. And we see in humans they have this crystalline structure here. This is the ranchicrystalline structures that we see there. Here we can see the elongation of some of the spermatids, and finally they become very dense and concentrated, as you see there. So if we go back and go, next one we're going to see is the toluidine blue stain one, and here this one shows you the reedy testis tubule. So some of the tubules are here. They discharge in the reedy testis tubules, go out through the efferent ducts and through the ductus epididymis. So we'll be looking at this region right in through here and look for the fusion of the seminal tubules with the reedy testis tubules. Okay, and so we take this one, we'll take a look at this testis, and when it pops up, we'll be able to see it. Okay, so I'm going to move this a little bit, And lo and behold, this is the reedy testis tubules right in through there. And so these are the seminary tubules, and we can see a similar type thing as what we saw before. But here everything is blue. The intensity of the stain indicates the density of things, but you have to look at the intensity of stain as well as shape and location to identify the different cells. Here's the interstitium. We see a nice Leydig cell right in through here, and then we see the myoid cells surrounding the tubule, and we can see the different cells inside there. These are Sertoli cells, the pear-shaped cells, as I mentioned to you. These are just the nucleus of these cells. The cell extends from the base to the lumen, as I mentioned before. And then we have the spermatogonia, spermatocytes, and spermatids, which are kind of located in the pockets of these cells. So the Sertoli cell has the germ cells, supports them kind of in their pocket. If we look at the interstitium, we can see Leydig cells. Sometimes they may have 2 nuclei, but usually 1. And we can see lipid droplets in through there. And this cell here with the red granules, that's the mast cell. There's another one there and there. You can see these very nicely with toluidine and blue staining. So these are endothelial cells. There's a red blood cell right in through there. There's another endothelial cell. But then these guys out here would be fibroblasts. This is another mast cell, and Leydig cells, as we can see there. And one of the things we want to see on this tubule is the union between the seminary tubules and the reedy testis tubule. So if this is a seminary tubule right in through there, and it finally goes to just Sertoli cells, and then that fuses with this one, and now we are in the reedy testis tubule. The reedy testis tubules are different from this blood vessel. You see, these are endothelial cells or flattened cells, as opposed to these are cube cells or maybe low columnar cells. So we can move to the next one. We're going to see this is a EPON section, and it shows you the interstitial cells. It shows you the myoid cells is what we're going to see. Fibroblastic type cells, myoid cells, spermatogonia, and we can see those. There's a Sertoli cell in a monkey, but this is the human over here. See the Leydig cells, and so if we look at that view, click on there, we can see the image come on, and we can see the different little piece of tissue. I'm going to scroll up to look at the interstitium, and we can see very nicely, we can see the details of the interstitium. You can see these are Leydig cells, and through here there's a nucleus, there's a nucleolus, a lot of smooth cytoplasm there, which would be smooth in the plastic reticulum. We can't see it at this level, but that's what it is. And here we see the lipid droplet. These are fibroblasts located in through here. This is a little arteriole with some smooth muscle cells, another fibroblast located in through here. And then these guys here are the myoid cells. Some people call them peritubular cells that are located in there. But we can see more fibroblasts, endothelial cells, smooth muscle cells, and then these are the spermatogonia here, and these are spermatids, and we can see the acrosomic vesicle developing from these spermatids, and then the elongation to occur, and ultimately they will be released. So you can take this image and manipulate this image as you like, move it around, and be able to see the different structures that are in there. And here we can see the interstitium very nicely in through there, and then we can see these are endothelial cells, myoid cells, spermatogonia, spermatocytes. These are Pachytene primary spermatocytes, and then these are spermatids. You can see the acrosomic vesicle here. Ultimately, they will look like bullets that will be released in the interstitium. Here we see a nice elongated spermatid with mitochondria moved in around its tail. And here we can see the different stages of the cycle, identified 6 stages in the human, and the different germ cells, the spermatogonia, spermatocytes, all the way up to here, secondary spermatocytes, and then that gives you rise to spermatids. You have said acrosomic vesicle developing, elongation of the nucleus, mitochondria moving around the tail, and you have completion of spermatogenesis at spermiation. So this concludes the images of the human testis, and we will have images of other ones coming in the near future. Thank you.